Hey everyone, today we are talking about a real unsung hero inside our bodies. It's this amazing enzyme that gets to work the very second you bite into something starchy, like a piece of bread or a potato. Its name? Alpha amylase. But have you ever actually stopped to think about what's really happening in that moment? What's going on at the molecular level that kicks off the whole process of digestion? Well, that is exactly what we are going to break down today. The entire thing gets started with one key player, who's really the star of our show, alpha amylase, and its mission is to start breaking down all those complex carbohydrates. So, to put it simply, think of alpha amylase as the first response demolition crew for carbs. When you eat starch or glycogen, which are these huge, really complex molecules, this enzyme just jumps right in and starts chopping them up into smaller, simpler sugars that your body can actually use. So, how does it actually do it? How does it pull this off? To get it, we need to zoom way, way in and see exactly how this enzyme performs its super specialized job. Okay, so the target is starch, but starch isn't just one simple thing. Nope, it's actually made of two different kinds of glucose chains. First, you've got amylose, which is kind of like a long, straight string of beads. Simple. And then you have amylopectin, which is way more complex. Think of it like a tree branch with a main trunk and all these smaller branches coming off the sides. All right, let's look at how these chains are actually held together. On the left, you see amylose. It's pretty straightforward. The glucose units are linked by what we call alpha-1,4 bonds. But check out amylopectin on the right. It's a little trickier. It has those same alpha-1,4 bonds all along the main chain, but look at those branch points. They use a totally different kind of link, the alpha-1,6 bond. And believe me, this difference is absolutely crucial. So here's the puzzle for our little enzyme. It's faced with this complicated amylopectin molecule that has two totally different types of bonds. You've got the alpha-1,4s that make up the long, straight parts, and then you have those stubborn alpha-1,6 bonds that create all the branches. And this is where the magic really happens. Alpha amylase is an incredibly precise tool. It's like it has a pair of molecular scissors that are designed to cut only one specific type of thread. It goes in and it snips only the alpha-1,4 bonds. Those are the links making up the long, straight parts of the chain. And what about those tough alpha-1,6 bonds at the branch points? It can't touch them. It just ignores them completely and moves on. So what's the result? Well, the enzyme basically chops up the long starch chains into these much smaller, more manageable sugars. But look closely at what's left behind. Those core branch structures, the ones held together by the alpha-1,6 bonds, they're still intact. They actually get passed along for other different enzymes to deal with later on. Okay, so now that we've seen the how, let's zoom back out and talk about the where. We're going to follow alpha amylase on its pretty wild journey through the body. And it's not a one-stop shop, let me tell you. It's a multi-stage mission. This journey has three really distinct stages. It all kicks off in the mouth. Then it takes this unexpected break in the stomach. And finally, it completes its mission down in the intestines. All right, stage one. It begins the moment you start chewing. Your salivary glands release alpha amylase right there in your mouth. This is actually why if you chew a piece of bread for a really long time, it starts to taste sweet. That's literally the enzyme breaking down starches into sugar right then and there. But then our little hero hits a major roadblock. As the food goes down into the stomach, all that hard work comes to a screeching halt. Why? Well, the stomach is basically an acid bath, and alpha amylase just can't work in that environment. The acid completely deactivates it, putting the whole mission on pause. But don't worry, the story doesn't end there. As the food moves out of the stomach and into the small intestine, guess what? Reinforcements arrive. The pancreas releases a whole fresh batch of alpha amylase, and in this much friendlier environment, the new crew picks up right where the first one left off, finishing the job and breaking down any leftover starches. So, this is a really fascinating process, right? But why does it matter to you? How is this practical? Well, it turns out this hardworking enzyme is also a super important indicator for our health. Which brings us to a really important question. If this enzyme is supposed to be doing its job inside our digestive system, why on earth would a doctor ever test for it in your blood or your urine? Well, the answer is that alpha amylase levels in your blood can act like a really important warning signal. If those levels are high, it suggests the enzyme is leaking out from where it's supposed to be, either from the salivary glands or, and this is more common, from the pancreas. It's what we call a key biomarker that helps doctors figure out if something's wrong. And here's the main takeaway. One of the biggest reasons for this test is to help diagnose something called acute pancreatitis. This is a very serious condition where the pancreas suddenly gets inflamed, and seeing those elevated enzyme levels is a huge clue for doctors. So from being a super precise tool snipping molecular chains in your mouth, all the way to a critical diagnostic marker in a blood test, alpha amylase really is the first hero of digestion. Which just leaves us with one final thought. If this is just one hero, what other incredible biological heroes are working silently inside our bodies right now, keeping the whole show running?